How's it going, folks? Can you guys hear me okay? All right. Happy Wednesday. Welcome back. It is 12:01. We're going to get started. Any questions, folks? Any questions, any concerns, any issues? All right. So today we are starting new material, which is going to be covered in our next exam. And today we're going to talk about the special senses, and we're going to focus only on the eyes and the ears. Okay? Now, this is something that we have talked about before in the lecture, so it's not going to be new to you if you are taking the lecture as well. But we do have a lot of information that will be on our next exam. So I need all of you folks to start preparing for that next exam right now. You have seen the first exam, now you know the format. The format of the exams are exactly the same. You need to start looking at that exam list. You have an exam list, which is your study guide for the exam. Okay? You know what to expect now. You need to start going through that list. Okay? So let me share my screen with you. And let's talk about the special senses. Now, even though we're only going to focus on two of the special senses, we actually have five special senses, right? What are they? We have touch, we have smell, we have taste, we have hearing, and we have sight. Now, because we don't have time to cover all of them, we're only going to focus on the structures of the eye and the structures of the ear. So let's jump right into it and let's talk about the different structures of the eye. And guys, there will be several questions in the exam that is going to come from the slide right here. Now, of course, as we know, I'm not going to be using a cartoon for as a picture for the exam. I'm going to be using pictures of models, but the structures are still the same. Okay, you need to know all these different things that we're going to be discussing right now. And we know that our eyes have three layers that make up the eye. We have an outer layer, middle layer, and an inner layer. This outer layer is what we call the fibrous layer, and the fibrous layer is made of two different structures. We have this cornea, and we have the sclera. What is the cornea? The cornea is the front of the eye. It's the clear membrane in the front of the eye. And then the sclera is the white of the eye, which is going to cover, which is going to make up the entire eyeball, okay, with the exception of the cornea in the front. So together they make the fibrous layer. The middle layer is what we call the vascular layer. And it's called the vascular layer because we're going to find a lot of blood vessels running through this layer, providing nutrients and providing oxygen to the cells of the retina, the photoreceptors, okay? Now, the vascular layer is made of three different structures. We have what we call the choroid, and within the choroid, we're going to find a lot of blood vessels. We have the ciliary body, aka the ciliary muscle, because it is made of smooth muscle. And then we have the iris. And the iris is the colored part of our eye. And the function of the iris is to control the size of the pupil. What is the pupil? The pupil is the hole right here, okay, right through the iris. And the pupil allows the light to enter inside of the eye. Now, the iris does control the size of the pupil. It can either dilate the pupil, enlarge the pupil, or constrict the pupil. And that depends on the amount of light in the environment in which we are in. Okay? Now, if everybody look up here, please, we can see that the ciliary body has little strings that is going to connect the ciliary body with what we call the lens down here. This is the lens. And these little strings are what we call the ciliary zonules. And we're going to take a look at we're going to watch a video today of a cow eye dissection in which we're going to be able to identify several of the structures, including the ciliary zoning. Now, what is the function of the ciliary body together with the ciliary zoning? The ciliary body is going to control the shape of the lens. Guys, our lens, the lens of a living animal like us, it has the consistency of a gummy bear. It is not hard like plastic or like a piece of glass. No, it has the consistency of a gummy bear. You can squeeze it between your finger and you're going to change the shape of the lens. And the ciliary body does that. The ciliary body changes the shape of the lens depending if you're looking at an object that is close to our eye or if it is, excuse me, an object that is far away from our eye. Okay? This is what we call accommodation of the lens, and that is going to help to focus the light on the retina. Finally, we have the inner section of the eye, and this is what we call the sensory layer. And the sensory layer is made of the retina, which is the yellow section here that we're looking at all the way inside of the eyeball. Now, what is the function of the retina? The retina is where we're going to find the photoreceptors. And we're going to take a look at the photoreceptors in a minute. But the photoreceptors, they are responsible for the detection of light. They are what allow us to be able to see. Okay? What else do I want you guys to know from this slide right here? I want you to know the name of this structure right here. This is what we call the optic disc. Sorry, the optic nerve. What is the optic nerve? The optic nerve is one of 12 different cranial nerves that we have. And the optic nerve are all the axons of the neurons, of the photoreceptors that are exiting the eyeball. And those axons, they're going to bundle together in the back of the eye, and they are going to form the optic nerve. And the optic nerve is going to send information from the retina all the way to the back of the eye, a region that we call the visual cortex in the occipital lobe of the brain. Okay? So that is the optic nerve. In front of the optic nerve, we're going to find a circular structure right here. Right there where I circled. I need everybody to look up here. This is what we call the optic disc. Okay? Don't confuse the optic disc with the optic nerve. The optic disc is a circular area within the retina where all the photoreceptors, the axons of the photoreceptors are exiting the eyeball. They are leaving the eyeball. And in that spot, the exiting spot, where all the photoreceptors axons are going to leave the retina and leave the eyeball, there are no photoreceptors right here. The light cannot be detected right there because, again, there are no photoreceptors. It's only the axons that are exiting the retina. Okay? And this is why we call the optic disc the blind spot. And we're going to talk a little bit more about the blind spot in a minute. Another structure that I want you guys to know, everybody look up here, so you're going to be able to identify this in the exam. And here we have this little, slightly grayish, darker region of the retina right there, circled for you. This is what we call the fovea centralis. And the fovea centralis is the region of the retina where we're going to have the sharpest formation of the image, sharpest image formation. And the reason for that is because within the fovea centralis, this is where we find the highest concentration of photoreceptors. Okay? And because we have so many photoreceptors there, when the light hits the fovea centralis, we are going to see that image the sharpest. Now, when does the light hit the fovea centralis? When we stare straight at an object. Like right now, I'm looking straight at the camera. So the light that is hitting my camera and coming inside of my eye is hitting the fovea centralis. Now, can I still see with the periphery of my eye? Yes, I can, because I also have light coming outside of the fovea centralis, hitting outside of the fovea centralis on the retina. So this is what we call our peripheral vision right here. Okay? Now, is it going to be as sharp as when we stare straight at an object? No, but we still are able to see it. A couple other structures that I want you guys to know. We can see right here that in between the cornea and the iris, we have a space right there. This space is what we call the anterior chamber right here. And this anterior chamber is filled with a fluid called the aqueous humor. We're going to see this fluid as we watch the cow eye dissection. Now, in the back of the iris, we're going to find another chamber, another space, much bigger space. And this space right here is what we call the posterior chamber. And within the posterior chamber, we're not only going to find aqueous humor, but we're also going to find what we call the vitreous humor, which is a much thicker fluid. And this fluid helps to maintain the shape of the eyeball and maintain the retina attached to the back of the eye. Okay? So this is why we have our eyeballs they're filled with fluid. They're not air inside, it's actually liquid inside. Any questions, folks? All right. So moving on, let me talk a little bit more about the blind spot, and we can actually do a little test at home for the blind spot. You can see that, you can check that out if you want to. All that we need is a little piece of paper, just like what I have right here in my hand, in which on one side you're going to have an X, and a couple of inches away, you're going to have a dot right there. And what you do is that you're going to take this paper, you're going to close one eye, we're going to test one eye at a time, and you're going to keep this paper in front of your eye at arm's length. Okay? You're going to close one eye and you're going to stare at the X. Now, when I stare at the X, folks, right here, the light that hits the X and come inside of my eyeball is going to hit the fovea centralis because I'm looking straight at it. Now, can I still see the dot with my peripheral vision? Yes, I can. So now what you're going to do is you're going to take that paper, keep staring at the X, and now you're going to bring this closer to your eye. And it's going to be a distance from your eye in which the light that is hitting the dot and coming inside of your eye is hitting the optic disc, is hitting the blind spot. And you're not going to be able to see that object. You're not going to be able to see the dot. Okay? Then you close the other eye and you do the same with your other vision. Now let's talk about the photoreceptors that we find in the retina. We do have two different types of photoreceptors. We have what we call the rod cells or the rod photoreceptors, and we have what we call the cone cells or the cone photoreceptors. We only have one type of rod cell. They're all the same, but we do have three different types of cone cells. Okay? Now, the rod cells are involved with our dark vision, while our cone cells are involved with our color vision. Now, what are the different types of cone cells? We have what we call the blue cones, we have the green cones, and we have the red cones. Now, why are they called blue, green, and red? Do they look blue, green, and red under the microscope? No, they don't. The reason why they are classified as blue, green, and red, because these cells become activated, they become excited by different wavelengths of visible light. The blue cones are going to become excited by wavelengths of light within the blue range. The green cones will become excited by wavelength of light within the green range, and the red cones will be excited by wavelength of light within the red range. Okay? Now, some people they are colorblind. What does that mean to be colorblind? It means that they might have a deficiency in the number of one of the cone cells. It could be a decrease in the number of the blue, the green, or the red cones. And that's going to make it a little bit more difficult for those individuals to be able to see that specific color. Now, how are the rod cells involved with dark vision? The reason why the rod cells are involved with dark vision is because they have a very special protein called rhodopsin, and this protein is very sensitive to light. Even small amounts of light can excite the rod cells and can excite this rhodopsin protein here, which is something that doesn't happen with the cone cells. And this is why when you wake up at night in the middle of the room and it's dark in your room, you will not see any colors. You're going to be seeing shades of gray because with the amount of light that you have, it is enough to excite the rod cells, but it's not enough to excite the cone cells. Okay? Now, when the light hits the rhodopsin of the rod cells, they're going to split into two different proteins. They're going to split into what we call the opsin and the retinol, and that is going to lead to the formation of an action potential, which then is going to send information to our brain. So the brain can make sense of that information. Some individuals have what we call night blindness. What is that? That is a difficult time seeing at night. Okay? And the reason for that, oftentimes, not always, but oftentimes, has to do with the lack of vitamin A in their diet. Because vitamin A is essential for the formation of retinol. So if you don't have enough vitamin A, you cannot form retinol, which means you cannot form rhodopsin. Your rod cells will be defective. They're not going to become excited enough. So moving on, guys, here I have two simulated sites. One site is of a human being. The other site is of a cat. Okay. And this is also applied to any animal that is active at night. Cats, dogs, cattle. Can anybody tell me which one is the human site at night? The vision, the picture on the top or the picture at the bottom? Which one do you think is from a human? Top, top, top. Absolutely. So the simulated picture here from the top, this is representing the eyesight of a human. And then the one at the bottom here, this is representing the eyesight at night of a cat. And we can clearly see the difference here. We can see that humans can pretty much just see at night whatever is under the light right here. Anything that is on this side or around here, we can't really see it that. But if you look at a cat's vision at night, they have a much broader field of vision. Why is that? How is that different from humans? Well, let me tell you what cats and other animals have that we don't have. This structure right here inside of the eye called the tapetum lucidum. The tapetum lucidum. And folks, this is what we're looking at right here. This is a cow eye, okay? And I have done hundreds of cow eye dissections. And I have to tell you, the tapetum lucidum is the most beautiful natural color that I've ever seen in nature. You know, it has this iridescent, metallic, bluish, greenish coloration to it, and it's absolutely beautiful. I've never seen any color like that. Now, cats, they have this tapetum lucidum, and this is why they can see so well at night, because of this shiny, this iridescent surface of the tapetum lucidum is going to allow the light to bounce around in that membrane and hit the retina a second time, enhancing the vision. Okay. Now, cows, they also have the tapetum lucidum because they are active at night, and many other animals have that, but we human beings don't have it. And we use a cow eye as a model for the human eye. There are a lot of structures that we do share together. And what I want to do right now, I want to watch a little video with you guys that is going to show the dissection of a cow eye. Let's take a look at it.
Dream hair, real results. Let's start your journey today.
Today we're doing the cow eye dissection, and let's begin by looking at some of the external anatomy of the cow eye. We're going to start with the outermost structure right here, the most anterior structure, and this is the cornea. In an actual cow, this would be extremely clear like glass, but since this eye has been preserved, the preservative has denatured the proteins and caused it to turn cloudy. Around that, we see a lot of periorbital fat and connective tissue. And this connective tissue and fat plays an important role because this eye is located within an orbit, a bony orbit. And if you turn your head quick or you got a blow to the head and you didn't have this periorbital fat and you did not have all this strong connective tissue, then the delicate structures of the eye would easily be damaged. Let's look at the posterior surface of the eye, and we see this long tube-like structure coming out the back of the eye. That is the optic nerve, and that will go straight to the brain. Some of the other structures that I want you to see here is just some of this brown material here. See a little bit of brown material over here, and you can see pieces of it here where it's been cut, and also over here a little bit. Those are eye muscles. Those allow the eye to actually move up, to move down, to move left, to move right. Now let's go ahead and begin by removing some of this periorbital fat. So I'm just going to take the scissors and I'm going to attempt to cut some of this fat and material off, sort of clean up the eye a little bit so that we can see things a little bit better. It's starting to look a little bit better. And that looks good there. We'll move some of this material out of the way. And you can see the optic nerve much better now, coming out the back of the eye. We'll talk a little bit more about that in just a few minutes. We want to start the internal dissection by cutting the eye right just posterior to the cornea. Now, under some of this connective tissue is going to be another layer called the sclera, and we'll see that a little bit more clearly after we cut into the eye. So what I'm going to do here is I'm going to take a scalpel, and I'm going to hold it a certain way because I don't want the scalpel to slip. And this sclera that I was talking about, this material, is very tough to cut through. So I'm going to take the scalpel, and I'm going to hold it in the palm of my hand like this, so that the handle cannot move backwards. And I'm going to put my hands around it, just like that. And I'm going to take the scalpel. Be careful to have your fingers on either side of the eye. And I'm going to put it just posterior to the cornea, right around there. And I'm going to gently push straight down. You can see that the eye is, there's liquid inside the eye. And this liquid and jelly substance inside the eye helps maintain the shape of the cornea and maintain the inner shape of the eye. We'll take a look at that. When I cut through here, you're going to see some liquid actually come out. And that liquid is called aqueous humor. And that liquid, one of the things that liquid does is it helps maintain the shape of the eye. We certainly don't want wrinkles in the eye as light is passing through it. So let's go ahead. I'm going to push down gently, straight down, straight down. There we go. And you can see some liquid come out. You can see the liquid begin to come out. And all I'm going to do is roll the eye and gently cut while I'm rolling the eye. Be very careful of your fingers here. If you want, you can use a pair of scissors to also do this. I find it a little bit easier to use a scalpel. And be sure not to disturb the inner structures of the eye when you're doing this. And I'm going to open that up. I'm actually going to cut this right into 2 pieces. And let's take a look at the posterior aspect of the eye first, and then we'll look at the anterior part second. So I'm going to move this out of the way. And all the liquid that you're seeing right here, this is aqueous humor. The jelly-like material, that is vitreous humor, and it serves the same function. It serves to help maintain the shape of the eye. So we can see that there. What I'm going to do is I'm going to gently try to get rid of some of this vitreous humor, just kind of pour it out, nudge it out the side of the eye. And then I'm going to take a paper towel, and I'm just going to dry this up just a little bit so that we can see clearly in here. And we can see this cream-colored material that coats the inside of the eye. This cream-colored material is the retina. This material is probably the most important part of the eye, as it contains all the photoreceptors, the light detectors that actually detect the incoming light. Now, as I move this material, as I move the retina, you can see how I'm bunching it up and I'm moving it. It's actually going to be stuck to the eye at a certain point right here. We'll see this in just a minute. But one of the things I want to talk about here is what keeps this retina pressed against the inside of the eye. The vitreous humor actually does. The vitreous humor is going to press that retina against the inside of the eye. And when the vitreous humor is not present anymore, I can easily take this retina and I can just remove it and I can bunch it up. And you're going to see that when I bunch it up, it comes off rather easily, except in one place. In one place. And we're looking, there it is. You can see that one place right there where it appears as though the retina is passing through a hole in the eye. What that is that's actually the axons of the nerves in the retina. All the axons of the nerves in the retina are passing through the back of the eye. And where they pass through right here, the spot in the retina where they actually pass through is referred to as the optic disc or blind spot, because light cannot actually be detected there. Now, all these axons pass through the back, and if we look exactly opposite of that optic disc, that is where the optic nerve, all those axons are going to make up the optic nerve. That is where they exit and go to the brain. Now, going back to the eye again, that's the retina. The next part that we come upon is going to be this iridescent, metallic-looking structure here. Now, humans don't have this, but animals that are nocturnal, that are active at night, or animals that are corpuscular, active during twilight, early morning, late evening, that actually helps them see better at night. That actually reflects additional light and allows that additional light a second chance to strike the retina. Humans, however, do not have that. This structure is called the tapetum, Now, if I remove that, and actually the tapetum is going to be continuous with this other black layer you see here, that's called the choroid. The choroid is very vascular, contains a blood supply that serves the eye. The black color of this choroid also serves to absorb light. So when light comes into the eye, a lot of it is going to strike the retina and get absorbed, but extra light that doesn't actually that passes between the nerves of the retina is actually going to strike this choroid and get absorbed, so it's not reflected around the inside of the eye and we don't have blurry vision. Under the choroid is going to be a thick white layer. Now it's stained a little bit black from the choroid, but I'm going to get the forceps in here and you can see how thick it is. I'm going to peel back a little bit of the connective tissue here. You can see this very thick layer. That thick layer is called the sclera. And you can see as I push in on it, it actually springs back to its original shape. Very, very tough material, and that's why we had to hold the scalpel a certain way when we began to cut the eye. Now let's move on to the anterior part of the eye. And again, I want to remove some of that excess vitreous humor. So I'm going to remove some of that very gently. Don't want to disturb the other structures of the eye. So very gently, I'm going to remove some of that vitreous humor. And I'm going to take the paper towel and just very gently dab it a little bit so we can see clearly in there. You can see all that jelly-like material. Now, the first structure we notice is this gumball-shaped structure. This is actually the lens of the eye. This is the lens. It's responsible for what actually changes shape, and it's responsible for focusing the light on the proper place in the back of the eye. Now, again, in a live cow, or you, this lens would be very clear like glass. Now, this one's hard, but in a living organism, this lens would be very soft. You could actually squeeze it like you could a contact. Again, the preservative has denatured the proteins, made them cloudy, and made them very firm and rigid. Now let's take a look at this black structure that surrounds the lens. So you can see all these striations. That is called the ciliary body. And it is actually attached to the lens, and that ciliary body is going to be responsible for changing the shape of the lens. If we look close or if we look far away, that ciliary body is going to contract accordingly and change the shape, either flatten the lens out or make it more spherical like it is right now. I'm going to slowly remove the lens, and as I do, we're going to see if we can see where the ciliary muscle is actually attached to the lens. And you can see look, almost looks like saran wrap attached to the lens right there. You can see the attachment there. Those are suspensory ligaments.
These are also called the ciliary zonules. Okay.
And they attach to the ciliary body, and that controls the shape of the lens. I'm going to slowly pull that out, and you can see those suspensory ligaments tearing in two. And I'm going to remove the lens. So I'm going to remove the lens. If we were to cut the lens, it would look essentially the same. Might look layered inside, a little bit like an onion. So I'm going to move that out of the way. And we're going to take a look at some of these additional structures in here. Just going to move some of that out of the way. And medial to the striated substance was the ciliary body, is going to be a more smoother looking black structure. That smoother looking black structure, and I'm going to take the forceps and I'm going to grab it, is called the iris. That is responsible for allowing more or less light to enter the eye. During the day, you want less light, and at night, you want more light. So that iris is actually going to change shape, and it's going to allow this hole here, called the pupil, to become larger or smaller. That hole in the iris is referred to as the pupil. In cows, it tends to be oval, and in humans, it tends to be round. And then if I look through that, I see the cornea. That was the structure that we looked at initially right here. That's the cornea.
Any questions? All right, moving on. So we're done with the eye. Let's take a closer look at our ears now. And again, this slide here is very important. There are several structures that I'm going to be talking about right now that you guys are responsible for, and you will come across them in the exam. Okay? Now, just to remind you folks, we have two eyes, we have two ears, right? Both the eyes and the ears, they are bilateral. Can anybody tell me, what am I looking at right here? Am I looking at the right ear or the left ear? What is this one, Williams? Absolutely. This is the right ear, okay? This is going to be very important again for the exam and the quizzes. You need to tell me if the structure is bilateral. So every single one of the structures we're going to be talking about right now, they are bilateral. Now you might be asking yourself, what about the eye? What if I have just an eyeball, a picture of a model of a single eyeball sitting on a table? Can we tell if it is right or left? No. But if you have an eyeball that is sitting within the skull, so you have some references that you can tell if you're looking at the right eye or the left eye, then yes, you need to tell me if it is the right or left. So it depends on the figure. If it is a single eyeball, outside of the skull, sitting on a table, you don't have to worry about right and left. If it is sitting on the skull and you can see some references of the skull bones, then you do need to say if it is the right or the left. Now, the ears is divided into three different sections. We have the external ear, also called the outer ear. We have the middle ear, and we have the internal or the inner ear. Within the external ear, we're going to find what we call the auricle or the pinna. And the auricle or the pinna is this flap of a fat and cartilage that we have that we call our ears from here. And the auricle or the pinna, it serves as a funnel because it's going to funnel the sound waves that are coming from the environment. And it's going to lead those sound waves inside of what we call the auditory canal, also called the external acoustic or the external auditory meatus. Okay? And from the auditory canal, now the sound wave is going to travel all the way at the end, and then it's going to hit this membrane right here, which is also part of the external ear. And this membrane is what we call the tympanic membrane. Now, on the middle ear side, we're going to find the little bones, little ossicles. One of those ossicles, it is attached directly through the tympanic membrane. So when the tympanic membrane vibrates, because of the sound waves coming from the environment, now this little bone called the malleus will vibrate as well. And the vibration of the malleus is going to be passed through the incus. And then from the incus, the vibration is going to hit the stapes. Again, collectively, these are what we call the auditory ossicles. Now, the stapes is attached to another bone in the inner ear, and this bone is what we call the vestibule. Let me show you the vestibule right here. So the vestibule would be this section right there that I just circled. And inside of the vestibule is hollow. And inside of that space, we're going to find the fluid. And once the staples vibrate, folks, the fluid inside of the vestibule is going to start creating little ripples. If you ever take a stone and you throw it in the water, you can see the little ripples that is going to form on the surface of the water. Well, we're going to have these little ripples being formed inside of the vestibule. And these ripples, they're going to flow into what we call the cochlea, which is another bone of the inner ear. And inside of the cochlea, we have a little device called the spiral organ of Corti, and this is the device that allows us to be able to hear. We're going to take a closer look at it in a minute. Now, we can see that attached to the cochlea, we are going to find this nerve right here. This nerve is what we call the cochlea nerve. And the cochlea nerve is going to send the sound information to the brain. so the brain can make sense of that information. Now, attached to the vestibule, we're also going to find these arcs right here. These are what we call the semicircular canals. The semicircular canals. And the semicircular canals are also filled with fluid. And at the base of the semicircular canals, at the point where the semicircular canals attach to the vestibule, right here, this region is called the ampulla. And the ampulla is responsible for our dynamic equilibrium. We're going to talk more about static equilibrium and dynamic equilibrium in a minute. Now, behind the vestibule, we're going to find a different nerve that is attached to the vestibule right here. And this nerve is what we call the vestibular nerve. Now, both of these nerves, the cochlear nerve, which is attached to the cochlea, and the vestibular nerve, which is attached to the vestibule, they are going to fuse together as they are exiting the inner ear. And as they fuse with one another, folks, they become one nerve called the vestibulocochlear nerve. Again, the union, the joining of the vestibular nerve with the cochlear nerve. The last structure that I want you guys to know for the exam is this structure right here, this tube that is called the pharyngotympanic tube, also called Eustachian tube or the auditory tube. And this auditory tube connects our throat with the middle ear. And the function of the auditory tube is to balance the pressure inside of the middle ear. Now, very important, do not confuse auditory tube with auditory canal. If you call this one here an auditory canal, and this one the auditory tube, and this one the auditory canal, you will get that wrong in the exam. So be extra careful, okay? Now let's take a closer look at the cochlear region, where the cochlea is. And what I want to do, I want to take the cochlea, split it in half, and look inside. And what we're going to find, we're going to find all these chambers, all the spaces right here. And the spaces are filled with fluid. And we're also going to find a little device right there called the spiral organ of Corti. And we can see that the spiral organ of Corti is attached to the cochlear nerve, because the spiral organ of Corti is the region, is the structure within the ear that we are going to perceive sound, that is responsible for the formation of sound. Now let's amplify the spiral organ of Corti right here. And we can see the spiral organ of Corti contain this membrane right here. This membrane is called the tectorial membrane. And this tectorial membrane, as the little ripples that are in the fluid inside of the cochlea, they arrive at the spiral organ of Corti, the tectorial membrane will undulate, just like my hands here, guys. And as the tectorial membrane undulates, underneath it, we're going to find the little receptors called the hair cells. And these hair cells, they will become activated. They're going to generate action potential, and the action potential is going to travel through the cochlear nerve, sending sound information to the brain. Okay? Now, what about equilibrium? Now, equilibrium is going to be found at a different spot. We're going to see equilibrium is perceived in the vestibule and the ampulla. We're going to see that in a minute. But let's talk about deafness first. There are two different types of deafness. We have what is called conduction deafness, and there is what we call sensorineural deafness. What is the difference between the two? Conduction deafness is some issue with the conduction of the sound wave within the ear. So it could be an issue with the auditory ossicles, it could be an issue with the eardrum, or it could be because you have a big glob of earwax inside the auditory canal, right there, which is preventing the conduction of the sound wave. Now, the sensory neurodeafness, this is more serious. This is usually something related with the hair cells inside the spiral organ of Corti, or it could be due to a blockage or a damage to the cochlear. Okay. All right, now let's talk about equilibrium and let's see where equilibrium is perceived within our ears. And we do have two different types of equilibrium. We have what we call static equilibrium, and we have what is called the dynamic equilibrium. What is the difference between the two? A static equilibrium is our capacity to stand still and not fall over, okay, without moving, just standing up. That's static. Dynamic equilibrium is our capacity to spin around, to dance, to play, to ice skate, and then not falling off. That is dynamic equilibrium. And static equilibrium and dynamic equilibrium are perceived in different places within the inner ear. The static equilibrium is perceived within the vestibule, right here, folks. So if I take a vestibule and I open up the vestibule is hollow, just like the cochlea. And what are we going to find inside? We're going to find fluid. So there's going to be water inside of the vestibule. We're going to find the little hair cells. These are receptors. These are neurons, okay, inside of the ear. And we're going to find the little calcium stones floating on top of the fluid within the vestibule. And these little calcium stones are what we call the otoliths. Now, when we stand upright, just like what we're seeing here in the figure, folks, the otoliths, they have a horizontal position. And the little hair cells are standing upright. If I move my head forward, what's going to happen is that gravity will pull on the otolith, and the little hair cells will bend forward. The bending, the movement of the hair cells activate them, excite them, and they are going to generate action potential. And the action potential is going to travel through the vestibular nerve, sending static equilibrium information to the brain. Now, what about dynamic equilibrium? Dynamic equilibrium is perceived in a different spot. Dynamic equilibrium is perceived at the base of the semicircular canals in a region that we call the ampulla, right there. So let's do this. Let's open up one of these ampulla and see what we're going to find inside. What do we find inside the ampulla, guys? We are going to find the fluid. It is filled with fluid. So all of the bones of the inner ear are filled with fluid. We're going to find this membrane called the cupula, which is very similar to the tectorial membrane within the vestibule. And embedded in the cupula, we are going to find the little hair cells. Again, the hair cells. All right, let's do this now. Let's spin around. When we spin around, folks, the cupula, this tissue, this membrane inside of the ampulla, will spin in the same direction that we are spinning, either right to left or left to right. As the cupula spins, embedded in the cupula, we have the little hair cells, which are also spinning in the same direction. As the little hair cells spin, they become activated. They will become excited and generate action potential. And the action potential is going to travel through the vestibular nerve, sending dynamic equilibrium to the brain. And our brain then can compensate by contracting different muscles in our body, allow us to be able to stand upright, okay, and not fall. Any questions, guys? All right. So what are we doing today? Today we are watching two Edpuzzle videos. One of the videos is going to be a model. They're going to walk you through a model of the human eye. The second video is going to walk you through a model of the human ears. Remember, I use pictures of models for the exam. So you need to familiarize yourself with those models. After you're done with those 2 videos, and they are short videos, then you're going to jump on Lamaku and you're going to take the quiz for the lab today, lab 8. And then after we're done with that, you can simply log off the, okay? Let's get started, guys.